Hello and welcome, and now we're going to be delving into the world of genetics. So this week we're going to be talking about Mendel's experiments and heredity. So you can't talk about genetics without talking about Johann Gregor Mendel. He's kind of the father of genetics. In 1856 he made a primary model system for genetics with pea plants. And they were highly inbred pea plants, and he went by the concept of true breeding, where he used the same parents and the same offspring and continuously bred them. So there wasn't a lot of genetic variation there. He tested the blending theory of inheritance, where the original parental traits are lost or absorbed by blending in the offspring. And there's also the concepts that we should talk about of continuous variation and discontinuous variation. Continuous variation meaning that many genes determine a characteristic and they blend together. In humans, height takes a lot of different genes to determine. Discontinuous variation is when traits are inherited in distinct classes. There's no blending of traits together. And this is primarily what Mendel worked with discontinuous variation. So in his experiments, Mendel performed Mendelian crosses, or hybridizations, where he mated two true breeding individuals with different traits. For example, violet flowers with white flowers. The parental generation is known as P0. The first filial, or offspring, the first offspring of that parental generation would be denoted F1, and the second filial, or the offspring of that F1 generation, would then be F2. A trait is a variation in physical appearance of heritable characteristics. So he looked a lot at specifically the color of the pea plants to start. He self-crossed offspring of the same flower color, and he determined that 100% of the F1 generation had violet flowers when he combined violet flowers with white flowers. The white flower trait disappeared when violet and white were crossed. So, although 100% of F1 had violet flowers, when F2 offspring came into play, they had a mix of violet flowers and white flowers. So, this perplexed him because he thought that the white trait completely disappeared. But actually he stumbled upon recessive and dominant traits. So the violet color is a dominant color for these pea plants, and the white color of the pea plants is recessive. So the recessive genes can oftentimes be covered up by dominant genes, unless they're homozygous, which we'll talk about in a little while. In a reciprocal cross, this is a paired cross in which respective traits of male and females in one cross become the respective traits of female and male in another cross. So you can't really talk about genetics without talking about probability. Probability is the mathematical measure of likelihood. So to measure probability, the equation is the number of times an event occurs divided by the total opportunities the event can occur. And this is used to predict outcomes of Mendelian crosses. Traits can be expressed through genes on chromosomes. Alleles, oftentimes alleles are used synonymously with traits or genes, but alleles are gene variants that arise by mutation, and they exist at the same relative locations on homologous chromosomes. We also should talk about the difference between phenotypes and genotypes. Phenotypes are observable traits, and genotypes are their underlying genetic makeup. So a phenotype could be that you have brown hair, but the genotype would be the genes that make up or code for that brown hair. You can also have homozygous and heterozygous genes. Homozygous means you have two copies that are exactly the same. Heterozygous is when you have two genes that are not the same, that are coding for the same trait. So typically dominant genes are denoted as a capital letter, and recessive genes are coded as a lowercase letter. So homozygous, if you had two recessive genes, they were both lowercase for the same thing, that would be homozygous. If you had one big letter and one little letter for the same trait, that would be heterozygous. So a really good way of looking at this is through Punnett Squares, which you may have done in high school, right? They were derived from a gentleman known as Reginald Punnett. They help to predict outcomes of genetic crosses. So this is an example of those pea plants that we were talking about earlier. Yellow pea plants are dominant, green pea plants are recessive. So we can use the letter Y for each one to determine the color of the pea plant. Okay? So a capital Y is because the yellow is dominant, lowercase because the green is recessive. So for each Punnett square, you go through and combine the traits that meet at that square, or the genes that meet at that square. So big Y and little y meet here, big Y and little y meet here, little y and little y meet here, and little y and little y meet here. So you can see that the two top possibilities are heterozygous because they have a big Y and a little y. And because we know that the big Y, or the yellow gene for the pea plant, is dominant, it will cover up that recessive gene. So these top two possibilities will be yellow pea plants. The bottom two are homozygous because they have two copies of the same gene. They're both recessive. Therefore, these will be green pea plants. So there is a 50-50 chance of an offspring of these two parent pea plants. There's a 50% chance of the pea plant being yellow and a 50% chance of the pea plant being green. And you could do this with anything. If you don't know what your genotypes are, you can figure it out using a test cross. So test crosses can help to determine a dominant phenotype. If you know one parent's genotype for a gene, and then you have an unknown, but you know what the offspring look like, you can figure out what the genotype is of the unknown parent. An ethical way of doing this in humans is to do a pedigree analysis. Oftentimes this is done by geneticists to determine whether or not somebody is going to acquire a genetic disease. So they can figure out what the likelihood of passing that disease on to offspring would be before the parents decide to conceive. And usually this is done by a genetic counselor. Mendel determined from his experiments that two alleles exist for every gene, that alleles maintain their integrity in every generation, and that the recessive allele is hidden in the presence of the dominant allele. So, in the case of the heterozygous pea plant that we saw, it still carried that recessive gene for the green pea plant. So, those recessive alleles were carried, but they were not expressed. Some other variations are incomplete dominance, where the heterozygous phenotype is actually an intermediate between two parents. Sometimes when you combine two plants of different colors, they blend their colors to form a different color. So this could be a variation of a red plant mixing with a white plant to make a pink plant, and that happens sometimes with incomplete dominance. With codominance, this is when both alleles are simultaneously expressed. So this happens sometimes if you have an AB blood type, because the genotype for A blood is dominant, for B blood it's dominant, and sometimes when they combine they form AB blood. Many alleles exist at the population level. There are so many variations of genotypes and phenotypes, which allows for a lot of variation in species. The wild type is the most common phenotype or genotype. It's the standard or the norm. Anything else is a variant. Dominance hierarchies can exist where, while some genes are dominant, others can be more dominant or more widely seen. Some can be X-linked. So before all of this, we've been talking about inheritance patterns in autosomes or non-sex chromosomes. Now we're going to be talking about sex chromosomes with the X and Y chromosomes. So if a gene is X-linked, then it's on the X chromosome. In Drosophila or fruit flies, it's a little different, where they have one allele for any X-linked characteristic. So if you're a male and you receive the X-linked gene from the mother, then you will receive 100% of the recessive X-linked genes, and you will get the disease that is X-linked. However, if you are a female child of a carrier, then you will be a carrier. There are some special circumstances where females can get X-linked diseases. It's just very much less common than it is for males to get X-linked diseases. In order for women to get X-linked diseases, they have to receive recessive alleles from both parents. So the father has to have the disease, and the mother has to be a carrier in order for the female to get the disease. Some of the common sex-linked disorders are red-green color blindness. You'll see this a lot more in men. And hemophilia A and B, which are bleeding disorders. Oftentimes, the female child will go on to be a carrier and she won't know it, because if she received it from her mother and her father didn't have any traits, then she will not express any of those symptoms of the disease. Mutations may occur in essential genes and be passed on to offspring, leading to the offspring dying in utero or shortly after birth. There are two main types of lethal genes: recessive lethal, which is an inheritance pattern in which the allele is only lethal when it's in the homozygous form. So in the heterozygous form, oftentimes there will be an altered phenotype, something will be a little different, or they could appear completely normal. But if they're recessive lethal, then they're carrying that gene. Dominant lethal is pretty rare because these offspring tend to die off before they can reproduce and pass this lethal gene on to others. But in the dominant lethal, the allele is lethal in homozygous and in heterozygous form. One good example of this is the Huntington's gene in humans. And the reason this is passed on is because Huntington's has such a delayed onset. You don't know that you have it until you're about 40 or 50 sometimes. And by that time, you may have already passed that gene on to your family. So let's talk about some of Mendels' laws. He said that hereditary factors are inherited as discrete units, as genes. He had a law of dominance, which stated that one trait will conceal the presence of another. The dominant gene will conceal the presence of the recessive gene. His law of segregation states that paired genes must segregate equally into gametes, so that offspring have the equal likelihood of inheriting either factor. In his law of independent assortment, it states that genes do not influence each other. Every possible combination is equally likely to occur. And in a dihybrid cross, this is between two true breeding parents, and they express different traits for two characteristics. Linked genes violate this law of independent assortment because they do influence each other. Genes located physically near each other are more likely to be inherited as a pair. And we saw this in recombination and crossover when we talked about meiosis. Epistasis is another similar concept. So epistasis states that the interaction between genes is antagonistic because one gene masks or interferes with the expression of another, being dominant or recessive. And we can see this with pigmentation in mice.